the images you're looking at were taken with expansion microscopy, featuring our own expansion microscopy type, which is label retention expansion microscopy that gives you the high signal to noise ratio while retaining that super resolution. So I think we've seen the scheme for different scales of biological questions from cellular to protein level. And at the cellular level, diffraction limited microscopy should have no problem to help you to count the cells and tell which stage they are and how they connect with the ECM. But if you wanted to take a closer look at the subcellular levels, like the organelles, microtubules, axons, dendrites, you will need a super resolution, or even to the electron microscopy field to have like a sub-nanometer resolution to really see the conformation of the proteins and how they bind onto the drugs. And diffraction limits is around 250 nanometers, as educated in the previous talks by Fernando and other speakers. And so the previous talks were focusing on the optical approaches to super resolution microscopy, including all the methods, PALM, STORM, STAD, resolve, split, paint, SIM, structured illumination, and array scan, SOPHY. So these are already we're already talking about, and I want to focus on the second approach, chemical approach, which was started which was started by Ed Boyden lab at MIT around 2015, which is exactly the year after the Nobel Prize was given to the optical approaches for super resolution microscopy. And that was in the middle of my postdoc training. I was trained in Bohong's lab, and I saw a lot of Bohong's paper from your slides and images. So I was trained in the previous optical approach, but in the middle of my postdoc training, this approach emerged. So I quickly kind of redirect my research approach to the chemical approach featuring expansion microscopy. And the idea of expansion microscopy is basically a sample preparation method, very distinct from the optical approaches. You don't need an advanced microscope to achieve super resolution. Instead, you physically expand, enlarge your sample in 3D. So each dimension, you probably would expand your sample, enlarge your sample by 3 to 20 times. It depends on what protocol and what method you use. And I'm going to talk about it later. But briefly, it's just embedding your samples, for example, your tissues and the cells, or even virus and bacteria, in a swallowable hydrogel. And by adding pure water onto that swallowable hydrogel, the hydrogel will expand just like what's happening in the diaper. It's 99% identical chemicals as used in the diaper. And when the hydrogel swell, they will take the proteins isotropically to get apart from each other and swell enlarged together with the hydrogel. That's how the cells or tissues would be expanded in 3D. And to achieve, and this is the hydrogel. So they are kind of semi-firm, semi-runny, like 95% of water. But what's inside of the transparent hydrogel is your tissue and cells. They're at the bottom of the hydrogel, but still a part of the hydrogel. And of course, you should not put your hydrogel on the bench without a petri dish. This is just for show, but there are real tissues inside, but they are transparent. You won't be able to see them after expansion. And how expansion achieves super resolution? Of course, it didn't break the diffraction limit, just as the optical approaches, they didn't break the diffraction limit. They just have a way to get around with the diffraction limit. And here, the expansion microscopy resolution, XM resolution, is equal to the original resolution of your microscope divided by how many times you expand your gel in length. Like, for example, if you expand your gel in length or 10 times, then the resolution of a confocal would be reduced to 25 nanometers after the 10 times expansion on your sample. So here, when we talk about the expansion factor, we're always talking about the length expansion factor, not the volumetric. The volumetric should be like 100 to 1,000. And for example, this is the gel before expansion. After expansion, they're humongous, and sometimes they're too big to fit on your microscope. And then you need to cut them to image them. But after expansion, you usually can have a much clearer image under any microscope. Ideally confocal or area scan, because confocal has the optical sectioning, so you will see more clear when your sample is taller. But expansion microscopy at the same time is a sample clearing method. So you also, like, by preparing your sample, you already get rid of a lot of background noise. So even epifluorescence, you will see clear image. Okay, so how do we achieve isotropic reliable expansion of your sample? Of course, you don't want to induce any distortion of your sample. Otherwise, there's no point of imaging them after expansion. So to ensure isotropic expansion, we have the initial expansion microscopy developed by Ed Boynton lab has these 5 steps. First step is just regular immunostaining, optimized in your own lab for your own organelle or protein. And the second step is the add anchor step. So you add methacrylamide or acrylamide NHS ester to your sample. The NHS ester group will react with the lysines or any primary amines in your in the proteins. And what that does is to add a acrylamide handle onto the protein, which will cross-link onto the hydrogel in the third step when you start the gelation. So your proteins are anchored onto the polymer in your hydrogel. And the next step is homogenization. This term is kind of confusing. It's not really the homogenization as you do to extract the DNA or protein. Instead, it's just the digestion or a heat denature to break down the protein-protein interactions in the cells so that when they expand, they don't hinge each other to create that local distortion. So they're free to go. The neighboring proteins don't drag each other. So after the homogenization, they're free to be expanded and to be apart from each other. So the fifth step would be just adding the pure water for several hours and to allow the expansion to reach to its plateau. And then you will end up with a isotropically expanded tissue or cell sample. And here, as illustrated at the very end, these proteins, including the antibodies, they are further away from their neighbors compared to their initial position. So the sample is enlarged, and when you put them onto your microscope, they're further apart to be separated by the diffraction-limited resolution. And that's the original workflow. And over the years, there are another group of workflow, which is do the immunostaining after the expansion. So do the labeling after expansion. So here, the different thing is that when you do the homogenization, you can't do polymerized protein escape digestion. It's too strong. It will damage all the epitopes, which won't be recognized by the antibody anymore. So in this step, it's usually mild enzyme digestion or heat denature. So the epitopes are still protected at the end. Hopefully, you won't be able to protect 100%, but to some level, if your protein is abundant or antibodies are good, you can still stain them after the expansion. So the good thing is that your fluorophores are introduced later, so they are not quenched by the previous steps, but the limit is that you may lose epitope and your antibody couldn't recognize 100%. Okay, so that's the 2 workflows. And similarly to the optical approaches, there are a lot of acronyms of all different variations of expansion microscopy. Oh, I have one. Okay. So from the 2015 exam, and you have the Pro exam or exam with conventional antibodies, and later on is combination with super resolution optical approaches like XSIM or XSTAT, XStorm, and then it's combined with the lattice light sheet and 10 times expansion, iterative expansion. And in 2019, we developed our own version, label retention expansion microscopy, and the workshop will be next week led by my postdoc. And the relationship among all of these is based on the previous two workflows. So this will be stain first and then expand. And this will be expand first and then immunostain. So these are the two families we've been talking about. Okay, so one thing. On the bottom, you will see a processing bar for my talk. So you can expect what's coming. And after the pandemic, I noticed all my students have like very little patience if they don't see a processing bar. So this is for your guide. So next step, well, and there are many, many more expansion microscopy coming. I can't, well, appreciate more how hard my colleagues are working on these, and each of them has their own expertise. Feel free to take a photo of this so that you can trace the references. Next, I'm gonna unfold the LR expansion microscopy to tell you why it's necessary to develop such a protocol and what it's good for. Our goal is to combine the optical approach with the chemical approach to achieve molecular resolution. So we thought it's just a simple combination, but when we, and if we achieve the resolution, then we don't need electron microscopy anymore. Oh, well, that's just a joke. So we will break down into their regime, but still each method has its pros and cons. But we expect to have like a nanometer resolution when we combine storm with expansion microscopy. However, when we start to combine these two methods, we found a huge problem that is a challenge in the original expansion microscopy, that is the loss of fluorescence during the harsh chemical procedure. So about 50% of the labeling, the organic dye, will be lost after expansion. And the worst news is the best photo-switchable dye suffer more than 90% of fluorescence loss after the 4-day procedure. So that's definitely not what we want to see. So we started to understand which steps cause the problem and how we should address these problems. So it turned out the two steps are most harmful to the fluorophores. The first is polymerization. So the gelation step is a free radical polymerization chain reaction. So a lot of radicals and high energy molecules are going on in that solution. So they bombard the dye and permanently switch the fluorescent dye to their dark state. So they don't come back and they don't, you don't see them after expansion. And the second step that is harmful is the proteinase K digestion, the homogenization. So the proteinase K can accidentally chop the antibodies or proteins into pieces. So if that piece is not cross-linked onto the hydrogel, you'll wash them away. So you lose some fluorophores on those pieces. To address these two problems, we developed this new method, label retention expansion microscopy, short as LR-XM, to retain the fluorescence using trifunctional labels instead of organic dye. So we replace the dye with this label. So each of the label has three functions. One is the reporter, which is a small molecule, biotin, digoxin or azide instead of the dye. They are non-fluorescent, but they survive the polymerization, and they can be recognized, they can be fluorescently labeled by, for example, fluorescent streptavidin, fluorescent anti-dig antibodies, or fluorescent alkyne after all the harsh chemical steps to introduce the fluorophores before the imaging. So you don't need to worry about the quenching of the fluorophore anymore. Second group is the connector. So we need to bind this trifunction linker onto the target protein through either the antibodies or a ligand that can recognize your protein tags, for example, snap tag, clip tag, or halo tag. So this functional group, bring the trifunctional linker onto your protein of interest. And the third group is the anchor group, which is the acrylamide or methacrylamide, not directly cross-link this report onto the polymer, the hydrogel. So even if you lose the protein of interest, your reporter is still in the hydrogel and will record the position of your protein of interest when you image them. And this project started at UCSF when I was still a postdoc with Bo Huang Lab and collaborating with Yan Sipo Lab, his postdoc Qi Li, undergraduate student Arthur Tran, and then finished by my postdoc Andrew and student Yin and my chemist Zhi Po. Okay, so the workflow of LR XDAM is very similar to the initial workflow. The different there are only two different steps. The first one is at the trifunctional linkers in the immunostaining step. And the middle part are identical to the initial expansion microscopy. And we have additional step, which is a post-expansion labeling to introduce the fluorescent dye to the biotin deoxygenin or azide, so that the dye is in the system for imaging, but not quenched. And here we use Clapeyron-coated pits to validate our results. So the reason we pick the Clapeyron-coated pit is that their size is under the diffraction limit usually, and it's about 100 to 200 nanometers in diameter. And there are a kind of hollow structure. And then with the conventional fluorescence microscope, you won't be able to see their hollow structure, and each of the dots, okay, I should use my pointer. Each of the dots is one Clapeyron-coated pit. So they're just one dot without structure. And if you use the standard or initial expansion microscopy with the Alexa Fluor 488 conjugated antibody, you will see like these dots are further away from each other, and you start to see a hollow structure here in this one, but not everyone, because the signal to noise ratio is relatively low because of the fluorescence loss. And with our method, you can have a much higher signal to noise ratio and see more hollow structures or even discontinuous one that is like reflecting the labeling of these polygons. And also some budding ones, which are smaller sized Clapeyron-coated pits, which are emerging from the plasma membrane. And the signal to noise ratio is about 5 times higher compared to the initial expansion microscopy for the sample. And we have another approach. If you want to have really high resolution, like 5 nanometers, you will start to worry about the size of the antibodies. So here we developed the second approach, which is the protein tag approach of LR expansion microscopy that breaks the antibody limitation of the size or availability. So instead of conjugating our previous trifunctional linkers onto the antibody, here we use a ligand instead of NHS ester in this trifunctional linker. So the ligand could recognize snap tag, clip tag, or halo tag co-expressing on the target protein. And the step is similar to the antibody approach. It's just the first step. You don't need the antibody anymore. You need to express the protein tag in your cell. And this is basically to show you from the conventional image of lamina A. So here, the structure you're seeing is nuclear lamina, which is a kind of meshwork at the periphery of the nucleus. And it's a meshwork, but you don't see the meshwork at all in the conventional image because they're really small under the diffraction limit. But with the LR expansion microscopy, you start to see the meshwork on the side. Also, you can add the second color by combining with the immunostaining approach of LR expansion, and you'll be able to see their spatial relationship with each other. The red color you're seeing is the nuclear pore complexes. And what's interesting is these NPC, so each of the dots is one nuclear pore complex. They perfectly sitting on the in the holes of the network of nuclear lamina. And as a structural biologist, the first question you will ask yourself is why they avoid each other. With this resolution, you see them perfectly anti-correlate with each other in the image. Here, we're lucky to know the reasons, but for a lot of other new relationship we see, we don't know. And that's the starting of the project. And here, because the nuclear pore complexes are known to connect with the actively transcribed DNA, and the lamina is known to regulate the heterochromatin. So functionally, they have opposite function, and here structurally, they're arranged by our cells intelligent enough to avoid each other, so their functions and structures are correlated. And now seems like the method is working, so it's time to combine with the storm to see that molecular resolution. So here we use the clathrin-coated pits again as our model system. The reason is that it's not only a hollow structure, but also a hollow structure formed by the hexagons and pentagons. So with 5 nanometer resolution, you should be able to see these polygons. And so if you focus your objective on the surface, on the top of the clathrin-coated pits, and you label, so here we label with snap tag on clathrin A. So here the green dots are where the snap tag should be. So if we reach molecular resolution, we should be able to see single dots and forming this array. And if we focus the objective in the middle through at the cross section of the clathrin-coated pits, you should see a discontinuous hollow ring. Let's see, if we use storm alone with 15 nanometer resolution, we won't be able to see individual vertices of these of this sphere. The reason is the resolution is not there, and you need to have a resolution that is smaller than half of the distance between the two vertices, which is less than 10 nanometers, to be able to tell them apart. And with the help of expansion, we achieved a 4.5 nanometer resolution. This is the effective resolution, not the microscope resolution. The pits, and we see the discontinuous ring. Okay, so that approves the resolution. So here I summarize the advantages of the expansion microscopy, especially LR expansion microscopy. So it reaches 5 to 17 nanometer resolution. You see a range, not a single number. It's because when you expand your samples at a different expansion factor, or when you image your expanded sample on different microscopes, you will reach different resolution. And the brightness is also an advantage with our LR expansion microscopy and the background clearing effect. And also you will reach high imaging depth with super resolution. In the optical approaches, there's always like a checks and balance between the imaging resolution and imaging depth. And here you can have super resolution with high imaging depth up to 1 millimeter when you use 2 photon. And it's quantitative. So now I want to move to the application of these methods. So the expansion microscopy methods could be applied for different biomolecules, including proteins, which I talked about, and the DNA, RNA, which I didn't talk about. These are the images from our lab. These are the each of the dots is individual RNA, and these are telomeres in the chromatin. And these are multicolor protein labeling with the LR expansion microscopy. And the expansion microscopy can be combined with different optical systems like SIMS, STAD, STORM, PALM, lattice light sheet. So these are the images from other people's work. And in our lab, we are really interested in the nuclear lamina. One of the reasons is it's quite unique to image the nucleus with expansion microscopy because you'll have the deep imaging power with the resolution, which you don't usually achieve with storm or palm. So here we focus on the nuclear lamina, which is a meshwork I explained, and this is what they look like with the electron microscopy. The reason we're especially interested in lamina is if something is wrong with nuclear lamina, the patient will disease a system kind of syndrome, including heart failure, intelligence, delay of intelligence development, neurodegeneration, and cytoskeleton skeleton problems, simply because nuclear lamina is such a big organelle regulating gene expression and support the whole chromatin as the mechanical sensor and mechanical support. So like these images features all the lamin A mutation patients. However, when we grow old, when we are aging, each of us will experience deformation of our nuclear lamina, like reassembling, recapitulate to what we are seeing in patients, but in a much longer time scale. So we're processing the nuclear defects in our lifetime. So if we could understand it more or figure out a way to reverse the deformation of lamina, I suppose each one of us will live to 200 years. So that's the motivation. So again, we start with the structure, and this is after using the LR expansion microscopy, you'll have the 3D information. The color is coded with a Z, and you'll see the meshwork a lot clearer. And that's even closer to the SEM image. This is our LR expansion plus area scan. I want to especially acknowledge Enrico and his and also Michelle and their working staff or scientist staff, Lorenzo, who helped us to take the first image of area scan in combination with LR expansion with the nucleus. And again, you can see the spatial information between different organelles, and it turned out nuclear lamina interact with a lot of organelles. In addition to NPC, it interacts with nucleoli, ER, mitochondria, whatever you name it, since it's so big and it's connecting the nucleus with the cytosol. And we figured very unique and special correlation between lamina and ribosome biogenesis. But today I won't have time to talk about it, but we are submitting our paper. So hopefully you will see the story soon. And also we applied LR expansion onto the tissue. I think many of you are interested in the tissue and brain tissue. So if you use conventional microscopy 10x objective to image the astrocyte, this is usually what you see. And with LR expansion, same objective 10x, very crappy objective, you'll see much better signal to noise ratio. And with 40x, you can see how astrocytes embracing the surrounding, almost every single surrounding cells with details. And also we're interested in Alzheimer's disease, and their spatial arrangement of these cells near the amyloid plaque are very different from what you're seeing right now in the healthy cells. And again, you can see their Z information. This is the color coded with Z. And you can see the surrounding cells when you add more markers. And here, what you are seeing in gray is just DAPI. And you can also see in details how they interact with the other cells, for example, the neurons and axons, dendrites, and how the astrocytes connect to the synapse. And talking about synapse, this is still confocal image, and you can see good separation between the pro and post synapse, with just only four times expansion. And here I summarize the applications as well. So basically, you can achieve super resolution imaging of a protein complex, RNA, and chromatin with expansion microscopy. To be honest, currently all my students switch to expansion, and our storm sadly is collecting dust in the lab. I think one thing is that they don't want to handle the photo switchable dye and don't want to wait in front of their microscope for 10 minutes per image. And so it certainly has some advantage for expansion microscopy. And also I think they will start to use the storm again to achieve that molecular resolution. So a combination of expansion microscopy with the optical approaches will be the final solution for the whole multi-scale application. And other applications include organelle structure, organelle contacts, like the lamina case, and also imaging of intact thick tissue, because now we can image as deep as 1 or 2 millimeters with the photons. So you don't need to thin section your samples anymore. You can section your sample as 1 millimeter thick and use expansion microscopy to image them. And molecular resolution, of course, and also you can image the extracellular matrix and brain tumor and microenvironment to see the interactions between cells. And lastly, I want to talk about our dissemination project with Leona and a few colleagues back home at UCI and in Japan. So basically, our goal is to distribute, to first fulfill the 56 requests from 2 years ago about our probes. So we scaled up our synthesis. So we synthesized the trifunctional linkers in our own lab. And special thank to our chemists sitting at the end of the room, Zhipeng, to scale up in our lab for free distribution. And as you can see, for Southern America and Africa, we only have 4% of the requests from there. So that's why I'm here, trying to convince you to use the method. Like other things I can't guarantee, but definitely it's cheap, and the distribution of the special probes for the next 2 years are free. So my postdoc Yuan Yuan will take some samples with her to here. Leona wants to know, how do you want to get the reagent? It does require 4C shipment or carry on. If you want to carry it back home this time, you need to figure out a way to keep them cool. But we'll have a little bit extra for you to try. And you'll definitely try it here in the workshop. So we're like back home at UCI, we're doing the validation, and we want to really elevate the super resolution imaging capacity here in Latin America. And here we have our first joint workshop next week. And last but not least, I want to thank all my students. So Yin and Lorenzo, they contributed most of the images I showed today. And Yuan Yuan, my postdoc, will lead the workshop. And Sohyang is is a student taking all the tissue images, and generations of students and postdocs contributed to this project. And this is our dissemination team. I really want to give a shout out to them, and UCI colleagues and UCSF colleagues. And so our lab is hiring actively, looking for talented postdocs, graduate students, or even technicians. So if you know someone or you are someone, please reach out to me through the email or Twitter. And so we have our beach parties in Southern California. Maybe that's not too attractive here in not a video, but we have our lab retreat there every year. And these are my sponsors to the research. And with that, I'm happy to take any questions.